Good morning, everyone, and thank you very much for the organizer to inviting me. Yes, I'm here to present the geeks perspective in that thing, so probably a slightly different tack again, mostly because what we are doing is we are, so this is my financial declaration of financial interest. What we're doing is really being involved in clinical trials. And the two main type of clinical trials that are being run at UCL by some of my clinician colleagues are either linked to prematurity or birth asphyxia, generally because between the two of them they are actually representing the largest burden of neonatal death, both leading to possible high risk of CP, learning disabilities, etc. So the outline of the talk is I'm going to start from the perspective, as I said, of the geek or the engineering. And I'm just talking a little bit about preemies as well here. And here the questions are often what can we measure properly head growth, maturation of the brain, this type of question. While in HIE, we are really more interested in metabolic disturbances. How can we be more specific? And we've heard already some hints about how we can do it. So I'm going to probably repeat a bit of that. How HIE is related to perfusion, what are the perfusion deficits, and how can you make your perfusion imaging a little bit more reliable. And finally, that's really so far only in preclinical, but I wanted to share with you some of the recent advances that we are looking at using a pH type of imaging, which could be very, very useful in this case. So these were the questions. So then the solution from the engineering. Well, for preterm babies, if you're looking at growth and maturation, you might be interested in myelin fraction. You might be interested in advanced microstructure imaging, or very advanced image processing technique for very careful assessment of head growth or brain growth in this case. For HIE, well, we are mostly relying on MRS for assessment of anaerobic metabolism, or ASL for measurement of perfusion, as the two previous speakers also showed. But here I'm going to actually show you why probably a lot of people still cannot rely on these techniques, because it's actually quite difficult to do still in these babies. And finally, I'm going to just touch on amide proton transfer weighted imaging for pH mapping. So just at UCLH, just to set the scene, there are 4 scanning slots a week, so it's about 40 to 50 infants that are scanned a year. So we have the 3T on the same floor as the neonatal unit. So with an incubator, we can actually be very efficient. Effectively, it would be best to have the scanner inside the unit, and as you know, some other groups in London have done that for quite many years as well. So the different type of cohorts that we are looking at are either the preemies or HIE with different clinical trials. And I'm going to start with some of the preemies. So the problem is really from a physics perspective. Well, this is a 30-week-old baby, 40-week-old. There's a dramatic increase of the brain size and also of the brain of the construction of all the sulci gyri here. And of course, if you have a baby of that kind, you see already that it's grossly problematic at birth, but at 19-year-old you have this effective development. And how can we better actually define all of this? This has been done, and this is being currently done by a whole group of engineers and processing people led by Seb Orsolin and Andrew Melbourne, who is also here in the audience. And so they've developed this kind of adaptive neonatal segmentation, which is, if any one of you has tried to use automatic segmentation tools on babies, can be extremely, extremely challenging. This is not like your average T1 for dementia project. It's very, very challenging. So here based on on lots of different advanced technologies, including probabilistic segmentation, partial volume estimation, and all sorts of methods to reduce noise and artifact. You can actually get a much cleaner, if you want, segmentation here for the in this case of the gray matter. This I don't see here the CSF. For example, in this case, so it's actually, as you can see, the standard type of tool will actually completely miss some of these areas. And then moving on a little bit into more detailed assessment of the white matter. So you can do some standard diffusion tensor imaging, and then you'll get these very nice color maps, which eventually change when the brain matures, and the change in this is mostly due to the reduction of here the fractional of the fractional isotropy. The water diffusion becomes more less directional when the brain matures, while actually the white matter tracks here start to form, and this is actually well known from pathology. So one interesting thing is this really happens through myelination of the cortex, and the myelination of the cortex is actually not the brain doesn't actually gets all myelinated and mature all at the same time. It starts with the very long long tracks here from the posterior all the way to the anterior frontal sides, and then slowly extend through the temporal sides. So we need to have some maybe better method to just look at myelin. And a method that is coming straight from the research used in part in MS is of course a multi-T2 mapping. So if you are having here, this is a micrograph of brains here, you can see that you have different elements. You have the intracellular components, you have a very strong, a very huge mapping component here, and then you have a very small extracellular space. And so if you would do some quantitative T2, you could actually use the equivalent of an inverse Laplace transform. There's different ways of doing it to actually separate the T2 contributions from the myelin water, the intra and extracellular water, and the CSF, for example. So you're using a multi-echo, typically 90 degree followed by a series of 180 degrees, and you fit your T2s, and then you separate these T2s in multiple components. So this is how it looks. So this is quite a few echoes. I don't remember how many, if we can count here. But with this, you can actually separate and quite nicely estimate the myelin water fraction here in this case. And you can see in these two estimation here, you have in these these red dots are here highlighted these the amount of myelin water fractions that actually tends to actually increase in this case with gestational age. So this is potential really interesting novel biomarker to be used in infant work. Another advanced method is the multi-shell type of diffusion, and there's like many ways. I mean, you just go to the meeting from tomorrow and you'll see, I don't know how many ways you can actually analyze these. This is one particular one that has been developed by Dr. Gary Zhang in our university, and it's just looking at the anatomy. You can see that the axon, both the axon and the white matter, as well as some of the dendrites near and close to the gray matter, have this kind of either very packed and directional packing of the fiber, or they can be less packed but still none less dense. So he has taken a multi-compartment model, which is actually separating the total signal into both a cellular extra tissue signal, as well as in the tissue a double model to model both the intraneurite spice, which is actually using a sticks type of model, depending of either a plate of spaghetti uncooked, if you want to take a Milanese type of comparison, or a plate of spaghetti that you will hold up by the center if you're in the tissue. So you have the intraneural space, which are sticks, and then the extraneural space, which is also still inside intracellular, where you have an hindered diffusion, usually having some kind of diffusion tensor elongated due to a lot of concentration in the intracellular tissue of intrasol type of metabolites in particular. So if you have here your DTI conventional, so you can actually, this is on an adult, but you have the general dominant orientation. You can calculate the fraction anisotropy. Of course, with this data, once you acquire 2 shells, you can actually reconstruct the way you want. But then you get different type of information, in this case an orientation dispersion, so it tells you whether the neurons in this case goes in one direction or whether they goes in generally are dispersed. They're doing this kind of ball of sticks all going in all different directions. While here you have the neural density, and as you can expect, the neural density is kind of in an adult brain here is kind of higher in the white matter and lower in the gray matter. The dispersion clearly shows, however, that even even in the white matter, sometimes you are dispersed, sometimes you're not, and then you get the extracellular water fraction as well. So this has been used in several studies. I'm just showing here one study that was done as a collaborative project between UCL and the group in Geneva, with Peter Huppi's group, where they actually looked, in this case, at this novel biomarker in preemies, and it seems to be relatively interesting, very easily done in only a few minutes, which is something that is quite good. We don't have yet, I have to say, the multi-band type of approach that exists at MGH and some other group, but we're working on it as well. Okay, I want to mostly focus now on perinatal asphyxia, or HIE. The problem is with the clinical problem with that we heard already about is, in spite of therapeutic hypothermia, it's still a big problem. And if you look at the survivor, there's still a large number of infants that will die in the first day in spite of aggressive treatment. Many will develop CP or something, and 40% will have very significant, can have very significant problems, including deafness, blindness, epilepsy, you name it. So there's a need for a new paradigm in general, and the new paradigm is maybe from a research, rather, not only physics research or engineering research, is really about development of new therapeutic agents or combined therapies or individualized therapies maybe. And for this, what we need is biomarkers. And I mean, if just moving a little bit sideways from pure physics or things, is like if you have the translational pipeline. So this is the standard way of how you get from a molecule of interest all the way into the clinics. There's a lot of stumbling block around, and this is the area where we are usually mostly useful with an MRI scanner, because once you get to phase 3 clinical trials, nobody will pay for such an expensive system. But there are actually these stumbling blocks, and they are the so-called first gap in translation is how do you take a new treatment from the preclinical study into phase 1, and then how do you actually go from phase 1 into phase 3 to actually improve the prediction or reduce the cost so that you can actually have a phase 3 clinical trial that works. Well, one of the possibilities is usually using a biomarker, because if you can actually use biomarker that are the same in all these different gaps, what's happening is that you can learn quite a lot even from the clinical study and then put it back into the preclinical world to get a virtuous cycle. And so one of these biomarkers, of course, what has to do with an energy metabolism. So this is very simplified cartoon of energy metabolism. If you have a glucose which gets through glycolysis, gets transformed into pyruvate, and if you don't have a lot of oxygen around, what's happening is the pyruvate dehydrogenase will actually transform it in lactate. That's one of the way of getting energy in the cells once you deprive it from oxygen, which is the basic, in this case, the basic pathologic events in HIE. Now, what's happening as well is that the cell needs to survive. It needs to actually produce enough energy to continue to survive. And there's different cellular process. So we're talking about three main enzymatic reactions. So pyruvate dehydrogenase is one, and then you have creatine kinase, which is actually buffering the PCR to be able to create some of the ATP, or the ATP synthase, which really needs, through the electron transport chain, needs actually the oxygen to actually recreate the ATP via the ADP. So all of these are a little bit in balance, and basically, if you're looking at our different probes here, from proton spectroscopy, what you'd get is a lactate and NAA as a neuronal marker, mostly. You can have some hints about choline, etc. I'm not sure. Phosphorus spectroscopy, of course, get you an assessment of all the three here, this difference between creatine kinase and ATP synthase activities, while NIRS can, in addition to blood flow, also give you some idea about the electron transport chain in this case. I'm just going to focus on proton spectroscopy here. What's quite interesting is, well, we've heard about it. So a baby with a normal outcome normally would show something like this, with a relatively normal LAA and low lactate. In a severely developmental delayed babies, you will have a presence of a very large lactate peak. Well, the interesting thing is we also have our piglet model, which is also used in a similar type of system. It's a slightly higher field, but it's still the same. And if you look at the baseline or after HIE, you can actually measure a very similar type of pattern. So a very severe lactate in a piglet will lead to a very bad outcome. I mean, in general, the piglet will have a very bad outcomes because the experiment is terminated after 2 days. Yet, if we count the cell death, this one will show many more cell death. And that's what's happening. And just to show you a story about how we're doing with our research, as if you're taking Xenon, and one of the latest final studies before Xenon was tried in the clinical trial was done in our lab by Nikki Robertson and her team. And if you look, so this is just a study where you have your piglet and they all have HI. HI is actually controlled in our lab. We are performing the HI under phosphorus spectroscopy, so we can actually tune it to get exactly a reduction of the brain metabolism to where we want. And so you can actually compare normothermia to inhaled xenon or hypothermia with inhaled xenon. And you do the analysis, and looking at the lactate and over NAA, you can see that effectively once you have a combined xenon and hypothermia, well, you basically, here this is your lactate over NAA, stays really, really constant. And these were the data that were used so that we could actually get into the situation of having a relatively large phase 2 clinical trial here in London on the three biggest hospitals, where we have now recruited, I don't know, maybe around 100 babies. Is it around that, I think, where they are either getting the normal treatment or they are actually breathing xenon for a couple of days to prevent cell death. And when you're doing this, one of the main biomarker that we're using is indeed spectroscopy, simply spectroscopy. And just as a matter of awareness, spectroscopy is it's very nice, it's very good. it's incredibly difficult to use if you're not used to it, and difficult to interpret. If you have an unprocessed spectrum as it comes out of the scanner, it can look a little bit like this. Once you actually process it, it can be much cleaner. So how do you process it? Well, you do all sorts of filtering, etc. For us, we do everything offline with GMRI and the LC model, and then you can do a multi-peak fitting, such as the LC model, giving like this, so which will fit some kind of baseline. You will have a residue that is very low, and then actually fit each of the peaks independently. The point is, this is a short echo time, this is a long echo time spectrum. It doesn't make much difference. This is relatively complicated to assess if you don't have anybody doing it. I just wanted to put like a little bit of a warning here. But, you know, in clinical data, generally, you know, this is a normal spectrum and this is an abnormal spectrum, and you could just clearly see a huge amount of lactic in this case. And this was recently reviewed also, and these are a summary of all the clinical trials having used lactate over NAA, and you can see that you can actually get a very, very good sensitivity and specificity. So this is a meta-analysis of a lot of trials. So it's not only a simple individual points here. These are all about meta-analysis of clinical trials showing that actually it has a very, very high sensitivity and specificity for neurodevelopmental outcome at 2 years. Okay, now the interesting thing is sometimes you see a lactate over NAA, so sometimes you see lactate, sometimes not. Now the question is this just simply directly related? You don't have blood flow, you have lactosis, high lactosis or not? And effectively, I think to answer this question you need perfusion measurement, and here I'm just going to go a little bit into the presentation of ASL while it was just used by the previous speaker, so I'm just explaining what is it based on. So it's based on, you know, this is probably the first quantitative perfusion measurement technique, which was the Kety-Schmidt method, where if you have a patient, you get the patient to breathe nitrous oxide, and by tapping on the concentration here on in the venous and arterial concentration, you can measure over time, and you can see that the difference basically between the arterial and the venous concentration gives you the blood flow. So this is the very basic, the very basic methodology that is used even in ASL today. Now in ASL you usually have an RF labeling period, which is continuous or post-ASL. The first speaker hinted at the fact that these don't give exactly the same signal to noise, and indeed there is an SNR advantage of using continuous or pseudo-continuous over post-ASL. There is a transfer delay followed by at some point an imaging. And how does it work? So this is really interesting, this thing. So you can actually use a Castle or a P-Castle experiment, or you can use a post-ASL experiment where you would do your labeling as a block under the area of expertise, or the original sequence, which was the FAIR sequence. So in our case, we use this one, and to be very precise, what we usually do is a short and quick angiogram to actually exactly put the labeling pulse here on the arteries, always in the same place. Now, the biggest problem when you're doing this is, well, babies have a tendency to move. I mean, the very, very sick babies that are intubated and under sedation won't move, of course. But quite a lot of them will be scanned even without sedation, so we basically feed them and then you wrap them and then they're all cozy and fall asleep. Of course, when they get into a scanner and they start hearing the noise of the scanner, quite a few will wake up in the middle of the experiment. So the fact is, on most of the clinical scanner, you will just directly get an average. So this, if you think that all your acquisition have been a little bit acquired in that way, this is a little bit dramatic, but a baby can move along. once you're trying to do the subtraction of each individual and average those, you will get something that is quite messy. And here, I'll show you just two cases, which, with the images coming straight from the scanner, you will barely be able to have any diagnostic possible. While if you really take care about having some automatic alignment procedure, even in post-processing so far, as well as a outlier detection and rejection. So if once you've tried to align them and individual scans are still showing lots of motion artifacts, in this case you need to take this average out of the whole average automatically, and to avoid contaminating that. If you do this, you pay a little bit of penalty in terms of signal to noise, but it's not nearly as bad as it sounds. So if you're doing this, from this type of imaging, you can actually quite recover very clinically relevant type of ASL. And some of the studies that we've been doing have been to actually hear a comparison between two HIE babies. And we've acquired the data at multiple post-labeling delays. So in this case, it was just using a simple post-ASL with multiple post-labeling delays to measure and to see, when do we see the perfusion, when does the large signal here in the large vessel disappears. And as you can see, quite interestingly, in a lot of severe HIE patients, and I'm sure my two colleagues will agree with me, you quite often see a very high perfusion. And Ellen hinted at the fact that this might actually be related to the fact that the cell death is actually completely undergoing at this point in time. And if you compare, like a severe HIE will have the large blood flow in the vessel disappearing very, very rapidly, while a more normal baby will have this artifactual signal, which is not perfusion in this case. This is really the signal in the large vessel present for a long time. So generally, if you're using a post-labeling delay, you will need to use at least 2 seconds after you've labeled to actually be in a relatively safe zone, in this case, where these flow artifacts are not present anymore. And here, my student Magda will present a poster where she's actually looking at different cases, and in which we have done both perfusion as well as spectroscopy. And you can see that quite interestingly, the lactate over NAA and the perfusion, in the first instance, if you look at the global whole brain or the thalamic perfusion, have very similar pattern, which doesn't seem to be correlated in any way. That means you can actually have a very low lactate over NAA and a very wide-ranging amount of perfusion, while effectively for very high NA over lactate, it's also generally high perfusion, but you cannot actually say anything if you have only one or the other. So actually, this just tells you that we might need a little bit more, even in MR, than just a perfusion to understand what's going on. Just in the last 2-3 minutes, I will just talk about some of the novel development. And I'm talking about chemical exchange saturation transfer. So this is a completely different type of field. And so if you have some different type of molecules, in this case it's glucose, but it can be any kind of molecules, these molecules will have exchangeable proton. And what I mean with exchangeable is if you are actually applying an RF pulse on these protons, you will actually reduce the magnetization, and by exchanging this magnetization with the water, you will actually affect here the water peak. So this is the principle of magnetization transfer, but magnetization transfer is looking at myelinated water generally, or at the semi-solid pool. These are free. We are looking at the proteins or that are in solution, so they are not in a semi-solid pool. And interesting, so and the way to look at it is we're looking like in magnetization transfer. You actually you vary the excitation pulse, and you're looking at the water peak. And once you do this, you end up with a so-called Z spectrum. And the Z spectrum is something like this, but we are very close to the water peak. This is in ppm, between -4 and 4 ppm. And here on this side, you will see, which is where you have most of the amide protons, which are rapidly exchanging at 30 Hertz. You can see that in the case of ischemia, you will get a signal that is actually showing a low pH. So this is a pH map. This was the original paper by Peter van Zyl, who showed that even in a model of adult stroke, you can actually quite nicely look at the pH difference in these maps. And so here we're just showing in our piglet model. And so here you have pre-HIE, 1 hour post-HIE, 24 hours post-HIE data, where you can see that this is the piglet brain in one slice, and this is the pH, the pH bar. You can see that you start having some acidification here in most of the cortical part, which eventually will turn into a complete acid, regularize as this. So these two cases, are graded from very bad to a good outcome. So it seems that, quite interestingly, if you're looking just really early post hypoxic ischemic injury, you will see a signal indicating a change in pH. Now we've done that directly following phosphorus MRS. So with phosphorus MRS, you can actually look at the difference between the PCR and the ATP peak, or the PCR and the PI peaks. All of those are calculating the pH and the intracellular pH. And if you're looking at the intracellular pH and you correlate with our ATP value, you see that you get quite a good correlation. Now the difference is this one is actually non-localized. It's just the entire brain, and the data that I showed you before really show a nice a nice image, really, of pH. So I guess the take-home message is, generally, it's an interesting field for physicists or engineers to work in because it has lots of challenges. We are using, again, I'm just repeating, these brain growth. We're using advanced imaging techniques to assess brain growth, as well as advanced diffusion-weighted imaging and quantitative T2 for maturation for asphyxiated babies. Our best biomarker of outcome so far has stayed proton MRS, but we are actually looking at many more. And I wanted to acknowledge all the people who gave me the slides. So the group here from Sébastien Ursulin, Gary Zhang, Daniel Alexander, Petra Huppi, as well as the group working on HIE, Nikki Robertson being the PI, Neil Marlowe is the PI on the preterm, and all the people here.